Hi, my name is Amy Callan, and I am a postdoc scientist at the Max Planck Institute for Evolutionary Anthropology in the Department of Primatology. Together with my colleagues in the Pan-African program, we investigate the drivers of great ape behavioral flexibility and diversity. For this publication, we collaborated with multiple researchers and NGOs across Africa to collect camera trap data from 14 field sites of wild chimpanzees, bonobos, and gorillas. Importantly, these data represented the first time these wild great apes encountered such a device. Therefore, the camera trap acted as a novel object in their natural habitat and simultaneously recorded their behavioral reaction to it. This is significant since novel object experiments are often used to assess temperament and personality traits such as exploration and neophobia, meaning the aversion to novelty. To date, the majority of animal research on this topic has been done in captive settings, and comparatively less is known about novelty in wild animals. Therefore, we considered our camera trap data as a large-scale field experiment for examining behavioral reactions of apes to a novel object in their natural environment. What we were looking for in the camera trap videos was a response or reaction to the device, also known as a looking impulse. This is when an individual will clearly look at the camera trap, orienting its head and eyes directly towards the device, showing visual attentiveness and exploration, which we then infer to be an indicator of curiosity. We found group-specific differences among the three species in their tendency to react to the camera trap. More specifically, bonobos and gorillas showed stronger looking impulses compared to chimpanzees. When the apes did look at the device, we found further group-specific differences in their tendency to show neophobic behaviors. Bonobos reacted neophobic much more often than either gorillas or chimpanzees. Neophobic behaviors included startling and retreat responses, as well as alarm calls or displays. We found no differences in neophilic behaviors across the species, such as approaching or touching the camera. And in general, these overt behavioral responses were rare. Overall, the two pan species were most divergent in their behavioral reactions to novelty. Bonobos appeared to use a strategy of being curious but cautious, while chimpanzees generally pay little attention to camera traps. Meanwhile, gorilla groups behaved a little like both pan species, but were also the most variable. We found no species variation in the amount of time individual apes spent looking at the camera trap. However, young individuals looked longer at the device compared to older ones. Individuals also spent less time looking at the device when they were together with more group members and when they lived near a long-term research site. This latter finding suggests familiarity with researchers may have desensitized these apes towards novelty in general. Some of our results nicely replicated findings from captive behavioral research. However, in addition to species intrinsic differences, our study highlights the influence of social and environmental factors on animal curiosity and neophobia. Overall, the behavioral variation we observed among our closest living relatives, the great apes, suggests that ancestral hominins may have exhibited similar variation in neophobia and exploration, and this may have facilitated behavioral flexibility and innovation that was needed to adapt to ever-changing environments. In the future, we hope to investigate novelty responses of great apes in more detail and to compare this to more distantly related primate taxa. This will help us to gain further insight into the selection pressures favoring traits such as curiosity.